Hello, welcome to lecture number 4. We will follow now with comparison of molecular sequences. Comparison of molecular sequence may be performed in pairwise alignments using 2 sequences or multiple alignments. And why we need to use comparison of sequences? What for? Well, as far as we know, the organisms and living organisms that we know come from cells where all the information is written in DNA sequence. So, all the evolution of the complex organisms that depend on DNA sequences that goes through evolution have came from modifications, DNA modifications, and sequences become from a common ancestor that evolves to give rise to different lines of evolution. Those mutations that occur in sequences must benefit the organism, otherwise the species or those organisms don't survive. And we can compare sequences as far as they perform similar functions. Otherwise, we can't compare sequences because they're different genes, different proteins. So, we know that evolution and diversity is originated by mutations. What kind of mutations do we know? Well, mutations occur at DNA level most of the times during DNA replication or even in other DNA cell division. Mutations can be substitutions, insertions, or deletions. The substitutions, as the name says, it's instead of one nucleotide, you have another nucleotide. When you have the nucleotides that are pyrimidines, like cytosine and thymine, if you change one for the other, it is called a transition because it still remains a pyrimidine. The same occurs changing an adenine for a guanine because they're both purines. So they have the same size and they will have the same type of binding. So this is called transition. The moment that a substitution occurs between the groups purine to pyrimidine or pyrimidine to purine, then you call it transversion. Either if it is an adenine for a thymine or a thymine for an adenine or an adenine to a cytosine or vice versa. These substitutions that occur in DNA can cause a consequence in the protein because gene sequences, DNA sequences, are transcribed and translated into proteins. So if the result in the protein is silent, it's synonymous, there's no big change in the protein. But these substitution can cause changes in protein. These are called non-synonymous mutations. And these consequences may be as a missense or a nonsense. And these kind of mutations that have these kind of consequences in protein are the cause of some diseases. For example, sickle cell disease or amyotrophic lateral sclerosis. It's caused by a substitution that causes a missense mutation. A nonsense mutation is the origin for cystic fibrosis. Let's see an example. A simple example, like if we have a DNA sequence, and you know the DNA sequences is read 3 in 3 nucleotides to give rise to amino acids. So, if we have the codons the, rat, cat, ate, the rat, if you have a substitution in this sequence, substituting an E to a C, it can cause change, but you still can understand the sentence. But if you have a different kind of mutation called deletion, that is removing one nucleotide, or an insertion, that is the insertion of a nucleotide in the sequence, you have a change in the meaning of the sentence from the moment you have the mutation. And these deletions and insertions most of the times are caused by errors in DNA replication. These mutations, like deletion and insertion, have caused changes from the place where it occurs until the end of the sentence, like until the end of the transcription. So the protein will be all different from the place where the mutation occurred in the gene. Either in deletion or insertion, you can't recognize the sentence, the red cat ate the rat, correct? So this type of consequence is called frame shift. From the moment you delete the E letter, there is a frame shift in the reading, because the reading keeps always being read 3 in 3 nucleotides. The same as in the insertion. After the E, you have an insertion of a C that changes all the reading frame from then on. So these insertion and deletions, most of the times called indel, cause frame shift consequences in the protein. The substitutions, let's see an example. At DNA level, if you have a TTC codon, that in mRNA after transcription you have an AAG, it translated into the protein level by the amino acid lysine. If you have a substitution from the C into the T in the last codon place, you have a transcription into AAA, and it still codes for a lysine. So you have a silent mutation. If you have a change in the first nucleotide of the codon from a T to A, you have UAG, and this is a stop codon. So it's a mutation that is nonsense. It's non-synonymous and nonsense. It causes the appearance of a stop codon in the protein. If you have missense mutations, the consequences from the substitution into protein is missense, is non-synonymous, but it may be conservative if the change causes the appearance of a different amino acid, but from the same group. So lysine, that was a basic amino acid, it is changed into an arginine, still a basic amino acid. If you have a change, a substitution in the codon at DNA level, and you have its transcription and then translation into a threonine because it was changed the T to a G, then you have a non-conservative missense mutation based on this substitution at DNA level, because you once had a basic amino acid that has a behavior and causes the protein to have certain function and certain structure. If you change to a threonine that instead of being basic is a polar amino acid, then you have a big change. The most aggressive change is to cause the frame shift based insertion and deletion, or substitutions that cause the appearance of a stop codon. So some examples of mutations in order for you to understand better. You have inserts and deletions that are called together as indels, because you comparing the sequence, sometimes you don't know if it appeared a new nucleotide or it simply disappeared. Only if you know the time of evolution of those species. Otherwise, you can't distinguish insertions from deletions. So at DNA template, you have by transcription into an mRNA sequence, and then read 3 in 3, starting always in ATG, you have the protein starting with methionine and then ends with a stop codon. If you have a mutated DNA that has an insertion of an A before the TT, when it's translated, it causes the appearance of an UAA codon that codes for a stop codon. So this protein is quite shorter. These type of mutations have a strong consequence. These nonsense consequences is one of the origins of cystic fibrosis, as I told you previously. But the worst consequences also is caused by substitutions, like Crohn's disease is caused by frame shift that is an insertion of a C at a position of 3020 in the gene that codes for a nucleotide binding oligomerization domain containing 2. This gene, this NOD2 gene, is changed, has an additional C at the position of 320, and it causes a change of the protein from leucine to proline at 1007 position, and then all the sequence is changed, and this is caused by a insertion. How can we see these mutations? We can see the mutations if we have two sequences of the same gene, two sequences corresponding to the same protein that has the same function, and we know it, and we can compare it. And we can compare only two sequences. These are called pairwise alignments. But how it will occur, how the alignments will be performed, this is the topic for the next lecture. Continuing lecture number 4. Thanks for your attention. Meet you soon.